Hello. Thank you very, very much again for the invitation. So just like Greg said, actually you didn't. So I'm a biologist, a neuroimmunologist by training. And I was always driven by biological questions. I was always fascinated by how cells make decisions. And at some point in my career, I decided to dedicate my research to study cancer. Where's the clicker? And my own personal view of cancer is this is a personal disease. This is something that I think affects everyone at some point, unfortunately. And this is what you see here. This is what I call my cancer family. And this is the reason that I decided to go in this particular path. And the sad truth is that we're not really doing as well as we should be doing. And despite of all the many efforts going into cancer research, if you look at the statistics in terms of death rate from cancer, the numbers haven't really decreased much over the last 40 years or so, as in contrast to cardiovascular disease, where there was a significant decrease in death rate. So the question is, why is that? Why is it such a big problem? And why haven't we been able to find the solution by now? So I think we need to start by talking about living systems in general, and to understand that we are dealing with astronomical complexity. We're dealing with millions of different entities, genes and proteins that are all interacting with one another, and everything happens in parallel. And this soup of chemicals is orchestrated in some manner using the inner cellular machineries to make sure that everything is happening according to some order and some plan, and cells divide when they are supposed to divide, and they die when they are supposed to die, and they move about when they are supposed to move about. And everything is normally under control. But sometimes, and this is quite rare actually, mistakes do happen, and suddenly cells start to proliferate in an uncontrolled manner. They lose their brakes, they lose their control system, and they stop listening to signals that tells them to stop proliferating, and they start to invade all the different parts of our body, and they nourish themselves with these blood vessels, and they reach a point where they are in some kind of a constant on state. And the problem here is that these are, by the way, the simplistic view of the hallmarks of cancer. And when we try to attack the problem, we really need to attack on all of these different fronts. It's not enough to just attack one of those. So this is part of the complexity. This is why it's such a complicated problem. But on top of that, we need to remember that cancer doesn't have any purpose, it doesn't have any plan, it simply evolves. So when you're dealing with an evolved process, the problem is that you can't really easily handle it and manage it and control it. And you can think of it as a hijacked physiology. That's the term I usually use, that we have our normal physiology and the way our cells are operating under normal conditions, and then suddenly they're hijacked to act like in a complete crazy way. So this is what we are dealing with here. And we need to work with that in order to be able to handle the disease. So what are we doing about this? So my research group has been pioneering over the last 14 years a new approach called the executable biology. And the way we work is that we think about biological programs and as computer programs. So we write biological programs as computer programs, and we use the same kind of techniques and methods that one can use in order to verify and check software and hardware to verify and check biological programs. And the idea here is that once you think about biology and biological processes as programs, you really need to explicitly figure out what is the sequence of instructions, what is the recipe, what is the algorithm that these cells are running, are executing in order to behave in a particular way. What is the recipe for proliferation? What is the recipe for migration? What is the recipe for cell death? Really explicitly write this like a recipe. And once you do that, you can then test these programs against the actual experimental observations that you gather in the lab. And you can automatically, in an unbiased manner, using these techniques, figure out the gaps between what we think is the explanation on the mechanism of how the system actually works and what we really observe in terms of the data. But the beauty of it is that it's super powerful because it does it automatically. It doesn't need a pen and paper, and we don't need to do it with our own head because this is quite a complicated mechanism to figure out. So this is what we do. This is the general gist of the approach. Now, obviously, if you want this to be used by clinicians, by biologists, these methods, these techniques have to be wrapped up in a package that really is very user-friendly, very biologist-friendly. I can say that because I am one, a biologist, and not that I'm friendly. And that there really need to be a catering for the fact that we are dealing with non-professional programmers. So as you can see here, we have such tools that are very graphical. You can drag and drop the different entities, whether these are cells or proteins, and you really construct these models in a very easy way. And even all the sophisticated analysis that we are performing on these models is shown in a graphical way. Now, over the years, we've been doing these kinds of models in various types of cancers, from blood cancers like leukemia, where we focused on the cell signaling, these soup of chemicals and their interactions inside the cells, all the way up to solid tumors, where we're modeling also the development of the tumors. And then we have a combination between the spatial aspect of the tumor and also the cell signaling. And in the work that we've done in leukemia, this is just one example, we were able to, using these kinds of approaches, we were able to predict new drug treatments or combinations that can then overcome a very common phenomenon, which is the resistance that patients acquire over time to a particular drug. So in many cases in cancer, we do have a solution, we do have a drug, but then over time, you remember, because as I said earlier, cancer is an evolved process, the cancer evolves around the drug, and then you get what we call resistance. So by doing these kinds of models, we can really get the 50,000 feet view and figure out all the different paths that eventually the cancer can evolve around, and make sure that we find the right optimal treatment that such a resistance will not occur. And more recently, we started collaborating with the pharma industry, in particular with AstraZeneca, and we use their clinical data, and we build these patient-specific leukemia models where we could really predict specific drug combinations that can reverse the effect of resistance. And then we went back and validate those predictions experimentally on the very same cell sample. So this is just to show you that there's this kind of approach really has some clear, meaningful clinical impact in the course of thinking in terms of personalized treatments. And of course, we are also working on more sophisticated models like what you see here. So in collaboration with neurosurgeons and radio-oncologists in the Addenbrooke Hospital in Cambridge, we are also modeling a very aggressive type of brain cancer called glioblastoma. And here the challenge is that there's a huge variability between patients, and you really need to tailor the treatment per individual patient. So the idea of these models is that once you have such a patient-specific model of the development of the tumors, and you can also integrate to it the genetic predisposition of the patient, you can then check the efficiency of chemotherapy and radiotherapy and how it will affect the growth of the tumor on a patient-specific level. So this is another recent effort. So obviously, I think you clearly see that this kind of executable biology approach has some clear advantages in on our fight over cancer. It leads to new biological insights and allows to identify new drug combinations and to reverse drug resistance. So the idea is that in addition to the many efforts that are out there to find new technologies and develop new means to do better detection and better diagnostics, by building these kinds of models of pieces of biology, and specifically in the context of cancer, thinking about the mechanisms, thinking about the behaviors of these cells as programs, trying to write them as cancer programs that can be executed by a computer, we can really go in the direction of getting a better understanding of how to control these mechanisms and how to control the drivers of the cancers. And that will allow us, this is the kind of work that will allow us to integrate all this vast amount of information into some meaningful understanding of how we can tailor the treatment in a much more better way generally, and also on a personalized level, of course. So my own good news, or a good take-home message that I have here, is that I think that even if we don't eradicate the disease altogether, I think by being able to control the disease and control the drivers, we can think about cancer as a chronic disease that you can live with. And I think at that point, this is quite a remarkable achievement. So that's my optimistic view. Thank you very much.